Today we will cover prokaryotic transcription and translation. Our learning objectives are as follows. We're going to look at the substrate for RNA polymerases, the requirement or lack thereof of primer, the direction of polymerization, proofreading ability, fidelity of the activity of the polymerase, and the speed at which transcription occurs. Transcription. This is RNA synthesis where ribosomal RNA, transfer RNA, and messenger RNA are being synthesized. This is different in prokaryotic and eukaryotic system. In prokaryotic system, there's only one polymerase that transcribes all of the RNAs, and polymerase is much simpler. In eukaryotic system, however, there are three types of polymerases. For instance, RNA polymerase 1 transcribes ribosomal RNAs 18S, 28S, and 5.8S. RNA polymerase 3 transcribes transfer RNA and the 5S ribosomal RNA. And RNA polymerase 2 transcribes the one that we're very familiar with, and that is messenger RNA. We're going to focus mostly on the prokaryotic polymerase. And the RNA polymerase of prokaryote is a 500 kilodalton enzyme complex that consists of two alpha subunits, a beta, beta prime, an omega, and a sigma subunit, so a total of 6 subunits. The holoenzyme consists of all of the 6 subunits, and the core enzyme is missing the sigma subunit. RNA polymerases. How does the RNA polymerase in prokaryotic system works? Well, the alpha subunits bind to the regulatory sequences in the promoter region of the gene that is going to be transcribed. And the beta and the beta prime subunits then bind to the DNA template and catalyze the addition of ribonucleotide triphosphate and the formation of the phosphodiester bond, and abbreviated PDP. The omega subunit stabilizes the RNA polymerase complex, and the sigma subunit recognizes the promoter, and it is the subunit that initiates the RNA synthesis. RNA synthesis consists of 3 stages. They are initiation, elongation, and termination. Initiation is a stage where the polymerase searches the genome, and it searches about 2,000 sites per 4 million base pairs. So it is very fast, and when it finds the site, it unwinds the template to form single-stranded regions, and then it selects the ribonucleotide triphosphate and begin forming the phosphodiester bond up to about 10 nucleotides. Elongation is after the formation of the first 10 nucleotides or oligonucleotide, then elongation occurs, and that is the sigma factor then dissociates from the core enzyme, and the core enzyme then proceeds through the open reading frame or the rest of the gene by adding one nucleotide after another. And then termination then occurs, that when the RNA polymerase comes to signals, and we're going to discuss these. There is row dependent and row independent termination, but when the termination signals are encountered, then the complex disintegrates and transcription ceases. Now, this is a one way that you can find whether or not a region, upstream region of a gene, is involved in polymerase binding, and that is that you take the DNA fragment and then you combine it with RNA polymerase and you treat it with DNase, and DNase will chew up the double-stranded DNA, and wherever the RNA polymerase binds, that region is going to be protected. And this is the basic principle behind what is called DNA footprinting, and that is that a probe or an upstream region of a gene that is suspected of binding RNA polymerase is radiolabeled at the 5' end with gamma p32, and then it is digested with DNase 1, and you know that when DNase 1 digested, it's going to randomly generate every possible nucleotide length, and wherever the RNA polymerase binds, it's going to show a protection on the sequencing gel. The labeling of the probe is usually done with an enzyme called T4 polynucleotide kinase, and it is the enzyme that adds the gamma p32 onto the 5' OH of the DNA fragment. Substrate can also be single-stranded, so it's either single-stranded or double-stranded. And normally, when you cut double-stranded DNA, the 5' end contains the phosphate group, and so in order for gamma p32 to be added to the 5' end, that 5' phosphate has to be removed, and this is removed by the activity of an enzyme called calf intestinal phosphatase, or it's abbreviated CIP. The core enzyme cannot initiate transcription, that it needs the alpha subunit to be combined to it, because the alpha subunit actually directs the RNA polymerase to the correct site where transcription has to be initiated. So the holoenzyme or the core enzyme cannot find the region where transcription must occur, but the holoenzyme with the sigma subunit bound actually looks or searches the DNA region, and it comes up with the right side through the activity of the sigma subunit. And this search is very, very fast. It is one-dimensional, not three-dimensional. And this is what this says, is that it is extremely fast. It is 100 times faster compared to the search if it were three-dimensional. And so the sigma subunit normally detects the region around the -10 and -35 for the RNA polymerase to bind. And so there are two elements that are usually found at -10. We call it the TATA box. TAT is the consensus sequence, and -35 from the start side for transcription is the sequence TTG ACA, and these are the two elements that are required for the RNA polymerase to bind, and the RNA polymerase is directed to this site by the alpha subunit, or not alpha but sigma subunit. There are a number of different sigma factors depending on the gene that is going to be transcribed. For instance, sigma 70 recognizes the consensus sequence that we've already shown, but sigma 32 is required for heat shock protein transcription. There's another sigma 54 is required when an organism is deprived of nitrogen, and it turns on genes that are involved in nitrogen assimilation, and the sigma 54 is required for the transcription of these genes. And so there's a number of sigma factors, and the the specific sigma subunit that is used depends on the gene that is being transcribed. All right, let's talk about RNA synthesis. RNA synthesis began by when the RNA polymerase finds the region where it needs to bind. There's a 17 base pair region that begins to unwind in that region, and this is facilitated by negative supercoding, and this corresponds to about 1.6 turns of the DNA helix. RNA synthesis proceeds in the 5' 3' direction. It is initiated with ATP or GTP, and it differs from DNA polymerase in that there is no primer requirement. There are 50 nucleotides added per second, and the 3' OH on the resident, and we're going to call it resident, 3' OH on the resident ATP then attacks the phosphodiester bond, and this is the alpha phosphodiester bond of the incoming NTP right here, okay, to form a chain that is now grown by one nitrogen base, extra nitrogen base. Okay, the fidelity for the transcription or for RNA polymerase is very, very low compared to DNA, and there's something that you need to think about why it can be tolerated. Why is it that low fidelity in transcription can be tolerated compared to in DNA replication? The sigma subunit dissociates, and then the core binding changes, and it begins to move through the rest of the gene. So initiation picture then is this, that RNA polymerase binds to the promoter region -10 and -35 right here, and this is the core enzyme containing the sigma factor, which is the hollow enzyme right here. And once it binds, it unwinds the DNA strands. The first nucleotide is added, and then the second nucleotide is added, and you get formation of the phosphodiester bond. Now you get the addition of nucleotide triphosphate until you get up to about 10 nucleotides on this oligonucleotide fragment that is growing. And every once in a while this is going to be aborted and you start back again, but once that 10 nucleotide oligo is stable, then the sigma factor dissociate and you go from initiation to elongation. And so elongation is when the bubble moves and nucleotides are added one at a time, and there is rewinding of the template for transcription behind it. So as the replication or transcription bubble moves, when the sigma factor dissociates, transcription bubble moves, and then there's rewinding of the DNA template behind. But there's unwinding in front. So there is rewinding behind and there's unwinding in the front. And then when it comes to a specific region where there is a formation of a hairpin loop right here that is self-annealing or self-complementary, forming a hairpin structure right here, then transcription then terminates. This is the row independent termination. So you've got formation of a region that can loop upon itself and form double-stranded region. When that is generated, then transcription ceases right here. Okay. Now, for the row dependent termination, there's a factor called Rho. This is a protein that actually binds to the RNA and it begins to travel and is chasing the growing, the beginning of the RNA right here. So this is single-stranded RNA right here. Raw protein begins to travel up, and when it reaches where the RNA and DNA hybrid is formed, it causes the dissociation of the RNA from the DNA template, and then RNA polymerase then dissociates, and the DNA template begin to rewind back. Eukaryotic transcription is different, as we pointed out. The transcription consists of 3 different polymerases. Polymerase 1 transcribes ribosomal RNA in the nucleolus, 18S, 28S, and 5.8S. And this is, you can differentiate between these polymerases, and that polymerase 1 is very insensitive to alpha-amanitin. Alpha-amanitin is a toxin that is found in some mushroom. Now, polymerase 3 transcribes transfer RNA and 5S RNA, ribosomal RNA, in the nucleoplasm. This is inhibited by very, very high concentration of alpha-amanitin. So it's not as sensitive to this toxin, but this toxin can also inhibit its activity, but at very high concentration. Now, the RNA polymerase 2 is the one that transcribes messenger RNA, and this occurs in the nucleoplasm as well. And this RNA polymerase is very sensitive to alpha-amanitin. So RNA polymerase 1 is insensitive to alpha-amanitin. RNA polymerase 3 is moderately sensitive to alpha-amanitin, and RNA polymerase 2 is very, very sensitive to alpha-amanitin, and that's how you can differentiate between the 3. Eukaryotic transcription again is different, because the gene for eukaryotes consists of exons and introns, while that of bacteria really doesn't have the introns that we see in the eukaryotic system. And so you have to transcribe the gene which consists of introns and exons into what is called a heterogeneous nuclear RNA. That heterogeneous nuclear RNA, that primary transcript, has to be spliced and connect only the exons to form the messenger RNA, functional messenger RNA. Again, this is a difference between eukaryotic transcription and prokaryotic transcription. Prokaryotic transcription, you have messenger RNA transcribed, and immediately protein translation can occur. While eukaryotic transcription, these are compartmentalized in that transcription occurs in the nucleus to form the messenger RNA. Messenger RNA is processed to form the polished messenger RNA, which is now extruded out of the nucleus into the cytoplasm, and there protein synthesis occurs. Of course, in prokaryotic system, you don't have compartmentalization, because the bacteria, of course, doesn't have nucleus, but you don't need to process the messenger RNA. As soon as transcription occurs, translation can begin. There are some processes, some techniques that allows us to study the role of upstream regions of a gene in transcription, and one of them is called the CAD assay, and this is chloramphenicol acetyl transferase. Now, CAD assay consists of the cloning of a gene for chloramphenicol acetyl transferase. Now, what this enzyme does is that it transfers acetyl group onto chloramphenicol. So normally what you would do is that you get a plasmid that has the CAD gene with no promoter. So it's got the CAD gene cloned into it right here, and then you can actually clone your promoter or the upstream of a gene that you suspect that might have any promoter activity upstream of the CAD gene right here. And then you transfect it into cells, and you let the cells express the chloramphenicol acetyltransferase enzyme, and you isolate the extract from the cell, and you begin to set up an assay. And the assay consists of extract of cells containing the chloramphenicol acetyltransferase plasmid, radiolabeled chloramphenicol, acetyl-CoA, and you spot these on thin layer chromatography right here. And so this is going to migrate. You're going to have unacetylated chloramphenicol right here, and then you're going to have acetylated forms of chloramphenicol. So the less the activity, the more that you're going to have unacetylated chloramphenicol, but the higher the activity, you're going to have acetylated chloramphenicol. So this is just one way of determining whether a upstream region of a gene binds RNA polymerase. This is another way of testing whether or not the region of DNA can bind protein for transcription, and that is that you can radiolabel a region that you suspect to bind protein, and then you interact it with protein extract containing, of course, RNA polymerase or transcription factor, and you run it out on acrylamide gel, and this is without any extract. The probe is going to run this fast. And if you add protein extract, and if this probe binds protein, it's going to retard the movement of the probe. And if you have an antibody, you can actually add antibody to this and determine what kind of protein it is right here that is bound to that upstream region, and it's going to retard the movement even farther. So if we look at the result right here, normally what you do is you do the assay, you run them on a gel, dry the gel down, expose it to film, and here just the probe alone, free probe right here. When you add the protein extract, it's going to have free probe, but a lot of it is going to be shifted upward because the protein is now bound to it. And if you add the antibody to it, it's going to shift it up even more because the antibody makes it even heavier than just the protein and the probe.